Hi, my name is Tim, and I will be talking about confocal microscopy. The technique was invented by Marvin Minsky, who developed it back in the late 1950s, and it hasn't been realized until modern times due to the invention of lasers. Lasers allow us to uh, use fluorophores, which are fluorescent molecules that can absorb light at one wavelength, and then they will use up some of the energy from the light, and then after it reaches a certain level, they will re-emit that light at a lower wavelength. So we send some light that is one color into the fluorophore, and then it will emit back another light at a different color. This emitted light from the molecules is then separated by a dichroic mirror, which is a mirror that only allows certain wavelengths of light to pass through it, and any other wavelength will just be reflected by it. In this way, we can focus on the emitted light of the fluorophore instead of any other wavelength that may cover up what we're trying to see. The two focal lenses are used to focus the sample image through a pinhole that is located at the focal point of the lenses, which gives the name the confocal microscopy. This pinhole prevents any of the background fluorescence from blurring the image, since when the laser is hitting the fluorophore, the fluorophores around it may also become excited and start emitting a light as well. However, those are not the molecules that we want to see. And so there's using these lenses, the focal point, they're not quite on the focal point of the lenses. So when the light passes through, they're without a pinhole, they're blurred to the camera. And so with the pinhole, we can block those light rays from traveling through the lenses at slightly different angles, and use the pinhole to just focus on the image we want to see. And in order to move the light around, in order to get a visual of the whole sample, we use scanning mirrors that can maneuver the light. And since not the entire specimen is illuminated at once, we use the scanning mirrors to move the light around to create a complete picture. The focal lenses can also be used to go deeper into the specimen, into the cell, and so we can get different layers along the z-axis of the specimen, and these different images along the z layer can be stitched together by a computer so that we can generate a 3D model of the specimen. Confocal microscopy is generally used to quickly scan tissue specimens. It can create thin optical sections of the specimen being examined, and so it can create a really quick thin slice of a cell or a muscle to see what proteins are active and other things like that. And then if we scan the entire specimen along the z-axis, we can create 3D images of the specimen that can be rotated around and enhanced so we can get a better image of the specimen. This is an example figure from a cardiomyocytes that of a rat that has been infected with beta-galactose, ADAP1, MEK1CA, and then ADAP1 and MEK1CA. And the whole purpose of this experiment was to see if ADAP1 and or MEK1CA was causing the increased formation of the alpha-actinin dense areas in these cells, in these cardiomyocytes. And the beta-galactose is the negative control, and so there's no areas of actinin dense puncta in the beta-galactose. ADAP1 shows a little bit in the cells where the white arrows point to the clusters, MEK1CA also shows a little bit, but when the ADAP1 and MEK1CA are put together, or injected together into the cell, then the result is there's a bunch of the actinin-dense puncta. The question I wanted to ask is, since confocal microscopy allows us to produce 3D images of a sample, can we use this technique to image extinct animal cells and use that to, I guess, visualize how modern cells have evolved from these ancient cells? And here are the references.